Developmental biology is a fascinating branch of science that explores how living organisms grow and develop from a single cell into complex beings. This field seeks to answer fundamental questions about life. How does a fertilized egg become a multicellular organism? What controls the formation of tissues and organs? By studying these processes, developmental biologists uncover the intricate choreography that guides life from its earliest state. This knowledge not only deepens our understanding of biology but also has profound implications for medicine, agriculture, and biotechnology. Let's embark on a journey through the stages of life, from a single cell to a fully formed organism. Every complex organism starts as a single cell known as the zygote. This tiny cell contains all the genetic information needed to build an entire organism. Through a series of carefully regulated steps, the zygote divides and multiplies, setting the stage for all future development. This initial stage is crucial as it lays the foundation for the organism's body plan and potential. The journey from one cell to many is a marvel of biological engineering, showcasing the power of life's blueprint encoded in DNA. After fertilization, the zygote undergoes rapid cell division, a process called cleavage. Each division produces smaller cells known as blastomeres without increasing the overall size of the embryo. This multiplication is essential for building the cellular foundation of the organism. The precise control of cell division ensures that each new cell receives the correct genetic material and is positioned appropriately for the next stages of development. This step is the first in a series of tightly regulated events that will shape the future organism. As cell division continues, the embryo forms a hollow ball of cells called the blastula. This structure is a key milestone in development, as it marks the transition from a solid mass of cells to a more organized, layered structure. The blastula's formation allows for the next critical steps, where cells begin to specialize and move to new positions. This organization is essential for creating the complex tissues and organs that will emerge later. Astrulation is a dramatic event in embryonic development. During this stage, cells move inward to form three distinct layers: the ectoderm, mesoderm, and endoderm. Each layer will give rise to specific tissues and organs. Gastrulation establishes the basic body plan, determining the organism's symmetry and laying the groundwork for the nervous system, muscles, and internal organs. This process is a turning point, transforming a simple ball of cells into a structured embryo. Once the basic body plan is set, cells begin to specialize in a process called differentiation. Guided by genetic instructions and signals from neighboring cells, each cell adopts a specific identity, becoming muscle, nerve, skin, or blood. This specialization is crucial for building the diverse tissues and organs required for life. Differentiation ensures that every cell knows its role and contributes to the overall function of the organism. Morphogenesis is the process by which cells organize into tissues, organs, and overall body shape. This stage involves complex movements and interactions, as cells migrate, fold, and connect to form the structures that define the organism. Morphogenesis is guided by genetic programs and environmental cues, ensuring that each part of the body develops in the right place and at the right time. The result is the emergence of recognizable features and functional systems. Organogenesis is the stage where organs begin to form from the three germ layers established during gastrulation. Each organ develops through a series of carefully coordinated steps, involving cell growth, differentiation, and morphogenesis. This process is highly regulated, as errors can lead to developmental disorders. Organogenesis transforms the embryo into a functioning organism, with specialized systems for breathing, digestion, circulation, and more. After organs are formed, the embryo enters a period of growth and maturation. Cells continue to divide, tissues expand, and organs refine their functions. This stage prepares the organism for life outside the womb or egg. Growth is controlled by genetic and environmental factors, ensuring that the body develops in proportion and reaches its full potential. Maturation also involves the fine-tuning of physiological systems, setting the stage for independent life. Development is orchestrated by genes, which provide the instructions for building and maintaining the body. Specific genes are turned on or off at precise times, guiding each stage of development. Mutations or errors in these genetic instructions can lead to developmental abnormalities. Understanding genetic control helps scientists uncover the causes of birth defects and develop new therapies for genetic diseases. While genes play a central role, the environment also shapes development. Factors such as nutrition, temperature, and exposure to chemicals can influence how an organism grows. Some environmental effects are beneficial, while others can cause harm or disrupt normal development. Studying these influences helps us understand how organisms adapt to their surroundings and how to prevent developmental problems. Stem cells are unique cells with the ability to become any type of cell in the body. They play a vital role in development, repair, and regeneration. Embryonic stem cells are especially powerful as they can give rise to all cell types. Understanding stem cells has opened new possibilities in medicine, including regenerative therapies and tissue engineering. Pattern formation refers to the processes that ensure cells and tissues are arranged in the correct positions. This organization is essential for creating functional organs and body parts. Signals between cells guide pattern formation, helping to establish symmetry, polarity, and spatial relationships. Disruptions in these signals can lead to developmental disorders or malformation. Apoptosis or programmed cell death is a normal part of development. It helps shape tissues and organs by removing unnecessary or damaged cells. For example, apoptosis sculpts fingers and toes by eliminating cells between them. This process is tightly regulated to ensure proper development and prevent disease. Cells communicate using chemical signals that guide development. These signals tell cells when to divide, differentiate, or move. Key signaling pathways such as hedgehog, W&T, and Notch play crucial roles in coordinating development. Understanding these pathways helps researchers manipulate development for therapeutic purposes. Developmental biology also reveals how evolution shapes life. Changes in developmental processes can lead to new features or species. By comparing development across organisms, scientists uncover the evolutionary origins of body plans and adaptations. This field, known as evolutionary developmental biology or EVODevo, bridges the gap between genetics, development, and evolution. Scientists use model organisms such as fruit flies, zebrafish, and mice to study development. These organisms are chosen for their simplicity, rapid growth, and genetic similarities to humans. Research on model organisms has led to major discoveries about genes, signaling pathways, and developmental processes that apply across species. Human development is a complex journey that begins with fertilization and continues through birth. Each stage, from zygote to embryo to fetus, involves intricate changes and milestones. Understanding human development helps doctors diagnose and treat developmental disorders and provides insights into the origins of health and disease. Sometimes, development does not proceed as planned, leading to birth defects or developmental disorders. These can result from genetic mutations, environmental exposures, or unknown causes. Research in developmental biology aims to identify the underlying mechanisms and develop strategies for prevention and treatment. Some organisms have remarkable abilities to regenerate lost body parts. Studying regeneration helps scientists understand how cells and tissues repair themselves. Insights from these studies could lead to new treatments for injuries and degenerative diseases in humans. Developmental biology doesn't stop at birth. It also explores how organisms age and how developmental processes change over time. Understanding aging can reveal ways to promote healthy lifespan and prevent age-related diseases. Knowledge from developmental biology is transforming medicine. It informs the diagnosis and treatment of congenital disorders, guides regenerative therapies, and supports advances in reproductive technology. By understanding how the body builds itself, doctors and scientists can develop new ways to heal and restore health. Research in developmental biology raises important ethical questions, especially regarding stem cell research, cloning, and genetic modification. Scientists, policymakers, and society must work together to ensure that advances are used responsibly and ethically. Open dialogue and thoughtful regulation are essential for balancing progress with respect for life. Developmental biology also benefits agriculture. By understanding how plants and animals grow, scientists can improve crop yields, breed healthier livestock, and develop pest-resistant varieties. These advances help ensure food security and sustainable farming practices for a growing global population. Biotechnology harnesses knowledge from developmental biology to create new products and therapies. Techniques such as gene editing, cloning, and tissue engineering are revolutionizing medicine, agriculture, and industry. These innovations offer exciting possibilities, but also require careful oversight and regulation. The field of developmental biology is rapidly advancing, driven by new technologies and discoveries. Researchers are uncovering the secrets of life at ever deeper levels, from single molecules to entire organisms. The future promises breakthroughs in understanding, healing, and even designing life itself. Modern developmental biology relies on a wide range of tools and techniques. Microscopy, genetic engineering, and live imaging allow scientists to observe development in real time. Advances in computational biology and bioinformatics help analyze complex data, revealing patterns and connections that were once invisible. Developmental biology connects with many other fields, including genetics, physiology, ecology, and evolutionary biology. Collaboration across disciplines leads to new insights and solutions to complex problems. This interdisciplinary approach is essential for tackling the big questions of life and health. Educating the public about developmental biology is vital for fostering understanding and support for scientific research. Outreach programs, science communication, and accessible resources help people appreciate the wonders of development and the importance of scientific discovery. Developmental biology reveals the incredible journey from a single cell to a complex living organism. Through coordinated processes of growth, differentiation, and organization, life unfolds in all its diversity and wonder. The study of development not only answers fundamental questions about life, but also drives advances in medicine, agriculture, and technology. As we continue to explore the secrets of development, we gain new tools to improve health, solve problems, and appreciate the beauty of life itself.